In this video, we're going to talk about ATP synthase. That's the enzyme that harnesses the flow of protons down their concentration gradient in order to make ATP. So we have a coupled reaction here. We have a very exergonic reaction of protons moving down their concentration gradient coupled to a very endergonic reaction of ADP being converted into ATP. So let's see how ATP synthase pulls this off. So for enzymes that are typically require investments of energy because they're normally the reactions are normally endergonic, that investment of energy is usually in the form of or necessary to convert that substrate to a product. Whereas ATP synthase, take a look at this. Here's the energy of our substrate, ADP, inorganic phosphate. And here is the energy of the product, ATP. Notice that it's actually exergonic, but also notice that it's not actually free ATP, that it's ATP bound to the enzyme. In fact, the enzyme bound so tightly to that ATP, that's what's driving this reaction. That's what's driving ADP, inorganic phosphate, spontaneously becoming ATP. The problem is that the enzyme binds so tightly to the ATP, to release it, we need to invest a ton of energy. And it's this investment of energy, this is the job of the proton concentration. When those protons move down their concentration gradient, that's the energy that allows us to unattach that ATP and free it from the enzyme so that we have free ATP. So let's take a look at the structure of ATP synthase. We have two main components, the F1 component and the FO component. Now the F1 component is all of these alpha and beta subunits, is this delta subunit right here that's kind of holding these alpha and beta subunits in place, this gamma and this epsilon subunit. Then FO is this C subunit, C10, and this A and this B2. All of this is embedded in the membrane, and it's holding this alpha and these beta subunits in place. So there is movement, though. And that movement is caused by protons moving down their concentration gradient from the intermembrane space over here to the matrix over here. And what moves here is this C10 spins, and it spins this epsilon and this gamma subunit. So this gamma subunit is kind of like a stick that's spinning within the alpha and beta subunits. Now, it's the alpha and beta subunits that are the important part here, the beta subunits specifically. Each one of these three beta subunits can exist in three different conformations. A beta empty conformation, a beta ADP conformation, and a beta ATP conformation. And each one of these three beta subunits cycles through those conformations. And the cause of them cycling through those conformations is this gamma subunit right here, this green thing. If we take a look at the top, looking top down at these alpha and beta subunits, if we look top down at this gamma subunit, we see that the gamma subunit is asymmetrical. It's not just a pole. It's a pole with something on the end of it. And when that something on the end of that pole is interacting with a specific beta subunit, it converts it into the empty conformation. So it was binding to ATP, but now it's in the empty conformation and it releases the ATP. As protons move through C10, they spin it which spins epsilon, which spins gamma, which moves this arrow here. And when that arrow moves, it changes the conformation of the beta subunit to empty. Now notice right here, anytime our epsilon is pointing to one beta, the other two are always in the other conformation. So this one here is in the ADP conformation, this one here is in the ATP conformation. And that's always going to happen. When it points to this next one, it's now in the open, the empty conformation, so it releases the ATP. The other one now has become the ATP conformation, and the one that was just emptied is now in the ADP conformation. So then protons move through, down their concentration gradient, through C10, spinning our gamma subunit, so that now that arrow is going to be facing the next beta subunit. That's now in the empty conformation, and ATP is released. And again, everything spins as protons move, and that arrow will just keep spinning over and over and over again. And with each movement, it releases a molecule of ATP. So we have a situation that looks like this. Starts there, ATP released as it's in the empty. This next one here is in the ATP conformation, but as we spin, it's now in the empty conformation. This one here is in the ATP conformation, but as we spin, it's in the empty conformation. And we just go round and around and around and around. And as we do, ATP is synthesized and then released from the enzyme. ADP is brought in, converted to ATP, released from the enzyme. And that all depends upon the energy of those protons moving down their concentration gradient, because it's not until that epsilon that it interacts with the beta subunit that ATP is going to be released. So what has been experimentally figured out is that for every 4 protons that is pumped out, okay, of the matrix into the intermembrane space, that is enough energy to create 1 molecule of ATP via ATP synthase. Thus, the 10 protons that are pumped into the intermembrane space from NADH, so 4 in complex 1, 4 in complex 3, 2 in complex 4, that means that NADH has enough energy to give rise to 2.5 ATP. And then for succinate, or FADH2, that pumps 6 protons. So the 4 from complex 3, the 2 from complex 4, which is enough for 1.5 ATP. So NADH supplies enough protons to be pumped out to make 2.5 ATP in ATP synthase, and FADH2 allows for enough energy, enough protons to be pumped for 1.5 ATP to be made via ATP synthase. And so now we can finish off our complete tally of the oxidation of glucose from glucose to 6CO2. And as we go through, we can see, okay, we're minus 1 ATP here, minus 1 ATP here, and then we're at 2 plus in glycolysis. So there's our net 2 from substrate level phosphorylation from glycolysis. We get a couple more ATP here from substrate level phosphorylation that occurs in the citric acid cycle. And then we have all of our NADHs, and we have our 2 FADHs here too. When you multiply that out by either 2.5 or 1.5 and then add all of that together, we get 30 or 32 molecules of ATP. The question now, what does this mean? Why is it 3 or 5? Right? It's talking about, it's in regards to the NADH that's in the cytoplasm that came from the citric acids, from glycolysis. Let's take a look at what this little dagger is saying. It says this number is either 3 or 5, depending on the mechanism used to shuttle NADH equivalents from the cytosol to the mitochondrial matrix. So that's something that we're going to look at in another video. What are those shuttles? How is these NADH equivalents getting from the cytoplasm to the matrix? But for now, our complete tally of ATP from the oxidation of glucose is either 30 or 32, depending upon how that NADH is being shuttled to the matrix.